In this video, we're going to learn about the structure and function of pigments. So first of all, let's talk about what we see and why we see what we see. So remember from grade 10 science, we learned about light and optics. And white light is a mixture of all colors of light. We can only see a small portion of the spectrum. And remember, if all the light is reflected from the surface, then this object will appear white. If it's all absorbed by the surface, it will appear black. And then if only red light is reflected by the surface, it will appear red, and so on. The thing is, the light that we see, the light that's reflected, is the color we see, but the color that is opposite that color is the light that was mainly absorbed. So if I saw something that was yellow, then the light that's being absorbed predominantly is violet. If I see orange, the light that's being absorbed is blue, and so on. There's two terms that you need to be able to differentiate. One of them is dyes, and one of them is pigments. So biological pigments are produced during metabolic processes, and they have the ability to absorb and reflect light and will appear colored. Dyes also will give things a desired color, but they're synthetically made to give things like food and fabric a desired color. So let's talk about the concept of chromophores. So covalent bonds can absorb light in the infrared spectrum, and electrons can absorb UV light and can jump to higher energy levels, but we can't see either of those because we can't see in the infrared or UV spectrum. So chromophores are molecules that have delocalized electrons, and those are able to absorb light in the visible spectrum, and that's what gives us color to our eye. So what is conjugation? So to be able to absorb light in the visible spectrum, materials have to have what is said to be a highly conjugated system. So what that means is they need to have multiple overlapping p orbitals, and generally that's 5 or more. And these molecules can be linear, like beta carotene, or they could have aromatic rings, like phenolphthalein. And we're going to look at a variety of different pigments and see why they reflect color back to us. It's because of those delocalized electrons. The first type of molecule we're going to look at is porphyrin. So what it has is a porphyrin ring, not a porphyrin ring, a porphyrin ring, which has 4 cyclic structures, and those structures have nitrogens and carbons within them. So you can see here, there's one of the structures. Just a second, I had the wrong pen. So here's one of the structures, here's another, there's the third, and there's the fourth. So when the porphyrin ring has functional groups substituted in, then it gets the term porphyrin. So porphyrin has 26 electrons in its conjugated system, so 26 delocalized electrons. So it makes it able to absorb and reflect light in that visible range. We're going to look at 2 different examples of porphyrins. The first one is chlorophyll, and the second one is cytochromes, predominantly or specifically hemoglobin, which is a cytochrome. So the first one, chlorophyll, the porphyrin ring has a whole bunch of substituted functional groups, and it has a central magnesium ion right here. This pigment appears green because it absorbs predominantly red light. And you can see all the different functional groups attached here all over the place. So it is much more complex than the simple porphyrin ring. And this magnesium ion is how you can identify that it's chlorophyll, because chlorophyll will have a central magnesium ion. Hemoglobin has a central iron atom. Cytochromes are porphyrins that have a heme, or a central iron atom, so right here at their center. So hemoglobin would be the example, and you can see all the different functional groups again. Cytochromes can undergo redox reactions, and that makes the iron convert from iron 2 to iron 3, and it can act as a catalyst in reactions. It also changes color, so it goes from orange red to blue red. And we'll talk a little bit more specifically about that in the next few minutes. So hemoglobin is a special protein. It has 4 subunits, so there are 4 polypeptides, and there are 4 heme groups. So each of those subunits has a heme. It's responsible for carrying our oxygen and our carbon dioxide through the blood between the lungs and the tissues, and we find hemoglobin in our red blood cells. We haven't heard about myoglobin before, but it's similar in structure to hemoglobin, except it only has 1 heme group. And instead of transporting oxygen and carbon dioxide, its job is to store oxygen until it's needed for cellular respiration. And you find myoglobin in the muscles. So, just a fun fact, when you see, when you have a sample of meat and you see like red material coming out of the meat, a lot of people mistake that as blood, and it's actually not. It's actually the myoglobin that you're seeing leaking out because the blood has been removed from the meat already, from the animal. So binding oxygen. Hemoglobin is a complex ion, and the oxygen ligand is acting as the Lewis base and the iron is acting as a Lewis acid. So when hemoglobin binds to oxygen, 1 electron pair from the oxygen forms a coordinate covalent bond with the iron, and the iron 2 ions get converted to iron 3. So that's where the redox reaction is happening. And then once 1 oxygen is bound, then it makes it much more easy for the remaining of the 4 heme groups to bind to oxygen more readily. And this action actually changes hemoglobin shape. So you can actually see the difference. This is the oxygenated version, and there's actually a deoxygenated version as well. So this diagram here is showing the oxygen saturation curve. So oxygen will move from an area of high pressure to an area of low partial pressure. So in low blood oxygen, there's not very much binding to hemoglobin. But as the blood oxygen levels increase, there will be greater bonding to hemoglobin until it becomes saturated. And then you can see that no more can be bonded to hemoglobin. And that explains the cooperative bonding. And we don't see that in myoglobin. So it doesn't follow the same shape. So there's a term called P50, and that's a measure, we use that to measure someone's overall health. So what that means is when 50% of hemoglobin is bonded to oxygen, and we should have a P50 of about 3.5. If it's higher than that or lower than that, there could be a problem. So that means that the curve will shift to the left or the right, but maintain the shape. So it'll still look like the S curve here, but it will shift left or right. There are many factors that affect the P50. So here's an example here. So the yellow line right here is showing our hemoglobin saturation curve at 37 degrees Celsius. So that's our normal body temperature. At 40 degrees Celsius, it's shifted to the right. So the reason why? So fever reduces oxygen's ability to bind to hemoglobin. So it shifts the curve to the right. If I increase the pH, it will shift it to the left. And if I decrease the pH, it will shift it to the right. When carbon dioxide levels are high, the pH drops, and that shifts the curve to the right. And then when carbon dioxide levels are low, the pH rises, and then the curve will shift back to the normal or beyond, to the left. So this diagram is quite helpful because we can see there's normal hemoglobin. We can see that myoglobin doesn't follow that S-shaped curve because it doesn't have that coordinate bonding. And then fetal hemoglobin, you can see that the curve is slightly to the left. Remember, the fetus doesn't breathe. It must get all the oxygen from the mother by the umbilical cord. And fetal hemoglobin has a higher affinity for oxygen than normal. And that's helpful because we need to be able to deliver oxygen easily to the developing fetus, and it doesn't want to have any challenges. So it actually is more readily binding oxygen than even the regular hemoglobin. And all of this hemoglobin, the fetal hemoglobin, gets converted or replaced by normal hemoglobin a few months after the baby is born. So it still will have some fetal hemoglobin, but it all gets replaced by normal hemoglobin. So carbon monoxide is a deadly gas, and the reason why it's so deadly is that it's colorless, it's odorless, and it's tasteless. So we don't know that it's even there and that we're being poisoned by it. And the reason why it's a problem is it's a competitive inhibitor for oxygen. So it will bind to hemoglobin and then block the binding site for oxygen. So then the hemoglobin can't carry oxygen. And another problem is it doesn't release as well from hemoglobin. So it remains bonded to hemoglobin even if there is oxygen available now. So the way we treat carbon monoxide poisoning is that people are given high levels of oxygen, so increases the substrate, the actual substrate to bind to, and eventually as the carbon monoxide is being released, then the oxygen then will be free to be bound to it again. Our next pigment that we'll talk about are anthocyanins. They're water-soluble pigments in plants, and they can be red, blue, or purple, and they're what give berries their color, but also their tart flavor. That's why blueberries, strawberries, and so on have a little bit of a tart flavor along with the sweet. And they're part of a group of molecules called flavonoids. And there are two fused rings right here, one of them being a phenol ring, so the A. Then those two rings are fused to another phenol ring. The conjugation that's involved here causes it to absorb yellows, oranges, and greens, which makes it tend to be red, blue, and purple. And because anthocyanins change their structure with the changing pH, it can actually affect the light that gets absorbed. So that makes anthocyanins really good pH indicators. So they tend to be red, pink, and acid. Then they become more purple, blue, and neutral, and then green, yellow, and base. So I don't know if when you were in grade 10 science learning about acids and bases, whether you had time to take a look at some of those natural pH indicators, but you can actually use fruit juices and vegetable juices to identify whether something is an acid or a base. So you can actually use red cabbage as an example. So if you extract the color out of cabbage leaves and then apply that to different pHs, you can actually make this entire rainbow that you see here. And that's just the changing of the structure of the anthocyanins, absorbing light in a different way and showing you different colors. It's kind of fun. You should try it. So metal ions have the ability to bind to anthocyanins, and that will affect their color. So this is kind of a fun thing. So when we can or preserve fruits and vegetables and we put them in cans, aluminum and iron ions that are in the metal of the can will bind to the anthocyanins, and that actually will reduce the color of the fruit or vegetable. So if you've ever noticed when you dump out a can of preserved vegetables or fruits, they sometimes aren't the nice crisp color that you get when you have the fresh fruit or vegetable. And that's why. Stability. So anthocyanins become less stable when they're in high temperatures and low pH. So high heat will break the rings, causing the molecule to lose its color. And that's why when you're cooking vegetables too, you'll see a decrease in the color, making it more colorless. Also, the low pH in our stomach, once we've eaten the food, breaks the molecule down and then it loses its color as well. Our next set of pigments that we're going to look at are carotenoids, and they're made by plants, algae, some bacteria, and fungi. And they're ability is to absorb violet, blue, and green, so they can be orange and yellow. And they can be absorbed and stored by the organism that eats them. So kind of a fun fact is if you eat too many carrots, for instance, or something with high levels of carotenoids, you'll store those molecules in your fatty tissues, and you can actually become a little bit orange, which is kind of an interesting thing. And sometimes people think they have jaundice, but it's actually they've eaten way too many carotenoids. Xanthophylls contain oxygen, and that gives them a yellow color, and carotenes don't, and that makes them more orange. So carotene is this molecule right here, and there's two different forms. There's alpha carotene and beta carotene. They're long hydrocarbon chains, as you can see here, and they have a ring at either end, and there's the beta form too. They're non-polar because of all these hydrocarbon units and no polar functional groups. So they're soluble in fatty tissues. That explains why the color builds up in tissues if you eat a lot of them. And this is actually what gives flamingos their color. So when they eat the food that gives them their color, they actually absorb it into their fatty tissues. We, like I said, can eat orange. We can become orange as well if we eat too much carotene. So photosynthesis. If you remember from biology when we learned about accessory pigments, carotenoids and xanthophylls and anthocyanins and so on aren't photosynthetic pigments. Chlorophyll is. However, these other molecules act as antioxidants. So they absorb light energy that would otherwise damage chlorophyll, and then they spread the energy around and help chlorophyll boost its energy absorption. And lastly, we can actually separate plant pigments by chromatography, which is what we did do in biology class. So depending on the solvent, different pigments will move further. So polar solvents will make polar pigments move further. Non-polar solvents would cause non-polar pigments to move further.